Hey guys, so our next topic is DNA replication, and the cell cycle. Today our notes are just going to be over DNA and DNA replication, so get that as your title, and we'll move on. Okay, the first thing I need you to remember from the last unit is a nucleotide. So if you'll remember, right, a nucleotide is the building block of a nucleic acid. And those nucleic acids function as your genetic information. So the three parts of a nucleotide, you've got the circle, which is phosphate. You get the pentagon, which is the deoxyribose sugar. And then you've got the rectangle, which is your nitrogen base. Now on the test, some of you guys just put that this was ribose. That'll work when we talk about RNA, but this time we're talking about DNA. And so that sugar has got to be deoxyribose. Also on the test, some of you guys said that this was just a nitrogen or just a base. You got to have both of those things, nitrogen and base. Yes, draw this picture in your notes again. Okay, label it again, because you will keep seeing it and keep seeing it and keep seeing it. Now, the whole reason that these things are important is because the specific order of them in your DNA, so mine's different from yours, okay, that's going to make you look the way you look, okay, so it's going to make you who you are. Alright, so nucleotides are always going to come in pairs, okay, so this is the way they pair. A is always going to pair with T, so adenine always pairing with thymine, and G is always going to pair with C. So guanine to cytosine. Yes, you need to memorize that. Yes, you will be applying that here in the next little bit. So A goes to T, G goes to C. Now, this scientist named Edwin Chargaff figured out that A bonds to T and G bonds to C. Basically, when he was doing all of this testing with DNA, he figured out, okay, well, there's an equal number of A's to an equal number of T's whenever I'm looking at my DNA structure, so I'm just going to assume, okay, that those two things pair together, and then eventually, whatever they figured out that those two things did, this guy won a pretty important prize. Okay, this is the shape of DNA. Okay, it looks like a kind of a spiral staircase. We call that a double helix. In 1953, two scientists named Watson and Crick discovered this shape of DNA. And then later on, a woman named Rosalind Franklin proved that that was actually a double helix by this technique right here, which is called X-ray crystallography. X-ray crystallography. Okay, so I just mentioned it. All right, I'll mention it again. The shape of DNA is a double helix. Okay, so if we were to untwist the twisty ladder of DNA, right, it would look like this. Now, the sides, which would be the gray part here, are your sugars and your phosphates. Okay, so for DNA, this will be deoxyribose and your phosphates. Now, the rungs, right here, where you would climb the ladder if it were an actual ladder, are those nitrogen bases. Okay, again, remember those four nitrogen bases are A, T, G, and C. Alright, now to hold those nitrogen bases together, we have what are called hydrogen bonds. Okay, so hydrogen bonds hold nitrogen bases. If you didn't have these hydrogen bonds, you would have a broken ladder, okay, which would mean your DNA wouldn't zip up correctly, and that would be a bad thing, you would think. So this here is just a picture to show you what the hydrogen bonds between those nitrogen bases actually look like. Okay, so in this picture you see the dots right here. Same thing over here, you get the dots holding those bases together. Alright, so DNA replication, what is it? When you replicate your DNA, you are producing two exact copies of that DNA. So you go from one, and then you're going to make an exact replica of the other. Okay, now this happens in the nucleus of our cells, and there's three reasons that we do it. Okay, we've got to replicate our DNA to be able to grow bigger. We have to replicate our DNA to be able to repair any broken cells, and then we have to replicate our DNA to be able to reproduce. Okay, some organisms can reproduce on their own. They don't need a mate, and so DNA replication is really important for those guys. All right, so there are 4 steps in DNA replication. The first thing that's got to happen is the DNA strand has to unwind. And then we have this enzyme, remember enzymes end in ASE, okay, called helicase, that's going to come through and it's going to hack or cut up those hydrogen bonds. Okay, remember those hydrogen bonds hold nitrogen bases together? So basically what's going to happen, helicase comes in and it splits those hydrogen bonds to be able to separate your DNA strands. So if you look right here, right, this was one strand and now eventually it actually has been split. Okay, then you get a second enzyme called DNA polymerase, and it's going to come in and paste in new nucleotides. So we're going to follow this blue strand here, guys. So if you see on the blue strand, there have been new nucleotides pasted in right here. On the red strand, that's polymerase that's doing that. Okay, the last thing that's going to happen, those strands are going to zip back up, just like down here, and we're going to end up with 2 identical DNA strands. All right, so that's it for DNA replication. I'll come back in a couple of days and talk to you about the cell cycle. See you later.